Hello students, today we are going to start a new chapter and that is chapter number 12 and that is applications of biotechnology. We have already learned about biotechnology and its principles. Now we are going to apply all principles of biotechnology in this chapter. Before we start the chapter, let me analyze this chapter topic wise. Let me see last 10 years analysis. Here you can see that is last 10 years analysis of a NEET examination from chapter 12. Here you can see list of topics from chapter number 12, that is a biotechnological applications in agriculture. You can see from that topic total 57 MCQs were asked in NEET examination. Next topic is a biotechnological applications in medicine. Okay, so from that topic you can see here total 24 MCQs were asked in NEET examination. Next topic is the transgenic animals. You can see total 8 number of questions were asked in NEET examination from that particular topic. And last topic is the ethical issues. From this topic total number of MCQs which is asked is 5. Okay, so you can understand by using these analysis you can get the idea which topic is most important. In previous chapter, we have already learned about tools and the principles of biotechnology. And today from this chapter, we will learn about how we can apply the biotechnological principles and tools. Okay, and in which field we can utilize these tools of a biotechnology. So we can apply biotechnological principles and tool in these particular fields. So here you can see this is 8 fields. Here it is listed over here. So first field is a biopharmaceutical industry. So here we can utilize or apply the biotechnological principles in biopharmaceutical industries. Then second is the therapeutic industries, then diagnostic industry. We can say these three is the medicinal purpose. Okay, means medical field this is. And fourth is a genetically modified crop. This is agricultural utilization. And fifth is a processed food. Then sixth one is environmental, that is a bioremediation. And seventh is a waste treatment project also. And last one is energy production. So here you can see there are a vast variety of fields we can utilize the biotechnological application. Here in this slide you can see there are 3 critical research areas of any biotechnological application, and that is to provide best catalyst in the form of improved organism, usually microbe or a pure enzyme. These all 3 are playing very important role to get good products. And second one is to create optimal condition. Without creating optimal condition you cannot get the good products. Okay, and through engineering for a catalyst to act. This is the second research area. And third one is to downstream processing technologies to purify protein and organic compounds. So these 3 areas are very important or critical research areas of any biotechnological applications. In today's lecture, we will discuss about application of biotechnology, especially in field of agriculture. How we can utilize the biotechnological tools and principles in field of agriculture. So today we are only discussing about agricultural application of biotechnology. Main aim of any agricultural industry is to increase food production. Okay, for that we have nowadays 3 options available. That is agrochemical based agriculture, in which chemicals are used as a fertilizers or pesticides. Second option which is available is organic agriculture, in which organic matters are utilized as a fertilizer. And third option which is available is a genetically engineered crop based agriculture. Okay, in which you can see genetically modified organisms are utilized as a fertilizers or a pesticides. Okay, and for that you can means use plants, bacteria, fungi, animals whose genes are altered by manipulation, which you have already learned in chapter number 11. Now let me discuss about some advantages of genetically modified organism, especially genetically modification in plants, that is also called GMO. So let me discuss about what is the benefits of a GMO. First is here it is written, that is it makes crop more tolerant to abiotic stresses. What is the abiotic stresses? Like a cold, drought condition, salt concentration condition, heat condition. These all are abiotic stresses, and if you have GMO in the field, then they are more tolerant to such conditions. Okay, second thing is the pest resistance crop. You can develop pest resistance crop which will reduce use of chemical pesticides. This is an eco-friendly method. Okay, so if you are using chemicals as a pesticides, that will act as a pollutant. Okay, so if you are doing this pest resistance crop, means if you are using pest resistant crops, then it will be very eco-friendly method. Then third one, it help to reduce post harvest losses. Okay, this is also very important. It increase efficiency of mineral usage by plants, and it will prevent early exhaustion of the soil fertility. And next one is it enhance nutritional value of a food. For example, golden rice, you can improve food quality also, crop quality also. We have developed already golden rice, which is we rich with the vitamin A, and that is also called vitamin A and rich rice. Okay, so what is a golden rice? This is not normal rice. This is a GMO rice. Okay, genetically modified rice. We have modified rice, normal rice, and we have now name given as a golden rice. What is a golden rice? Golden rice is the rice which is rich with the vitamin A. So this is very important. Now GM is used to create tailor-made plants to supply alternate resources like starches, fuels, pharmaceuticals. We can also modify the plants to produce starches, fuels, pharmaceuticals to the industries. So by this we can get the so many benefits of GMO. So first of all, let me discuss about the pest resistant plants. So what is a pest resistant plants? So pest resistant plants are acting as a biopesticides. So here you don't need to use pesticides which are chemical based pesticides. So if you are modified the plant in such a way which will resist itself from the pest, so it will act as a biopesticides. So pest resistant plant act as a biopesticide. It will reduce the need for insecticide. This insecticide may be a chemicals, so you can reduce the need of a insecticide. No more need of a insecticide, which is chemical based insecticides. For example, we have developed BT cotton, that is a Bacillus thuringiensis cotton, BT corn, BT rice, tomato, potato, soybean, etc. in such a field. Okay, it means these all crops which we have developed already are pest resistant plants or acting as a biopesticide plants. Now let me discuss about one example of a pest resistant plant, that is the BT cotton. So some strains of a Bacillus thuringiensis. This Bacillus thuringiensis is nothing but one type of a bacteria which is producing one kind of a protein that is responsible for killing insects like calypterans. Okay, that include beetles. Here you can see one kind of a beetle. Then lepidopterans. They can also kill the lepidopterans, that is a tobacco budworm or armyworm, and dipterans like flies and mosquitoes. Okay, and Bacillus thuringiensis form a toxin insecticidal protein. Okay, means this is the bacteria. This is the Bacillus thuringiensis bacteria which form a toxic, that is insecticidal activity, and this is one kind of a protein, that is insecticidal protein, and that is also called Bt toxin. This toxin type of a protein is developed from the Bacillus thuringiensis, and that's why it is called Bt toxin. That is one kind of a protein which is a crystal in form, and during a particular phase of their growth they will form this type of protein. It does not kill the bacillus, means it will not harmful for the Bacillus thuringiensis itself, and is it is existing as a inactive prototoxin. Inside the bacteria it will be existed as a inactive prototoxins. Now let me understand how this BT cotton is working. Okay, so first of all, when an insect inject the inactive toxin, as I told you, this protein is already prototoxin, not a active form, that is inactive toxin, and it is converted into active toxin. Whenever this caterpillar or insects are utilizing or ingesting such a kind of a crystal protein, that is a BT toxin crystals. So here you can see some parts of a plant will be ingested by the caterpillar, and this Bt toxin crystal will be inserted or ingested inside the organism, and now this crystal which is already inactive, okay, will be activated upon the solubilization inside the gut of a caterpillar or insects. Okay, and on the solubilization, what will that? It will be activated. Okay, so now it will be activated, and it will be, means as it activated, it will be received or bind on the receptor present on the mid-gut cell. Especially you have to remember, especially insect mid-gut cell have the receptor for the binding of such a toxin. Okay, now this toxin will be received by the mid-gut cells on the receptor, and after that what happen here, you can see some gathering molecules are there, some toxin oligomers are there. Okay, it will be react with the cells, and membrane insertion means they will insert inside the membrane, and afterward they will be generate some kind of pores, and these pores formation or a pore formation lead to osmosis of the cell lysis. Okay, as a result of that, cell will be lysed. Okay, and due to that cell lysis, you can understand cell will be now die. Okay, or septicemia may occur and larva or insect will be die now. Now let me repeat, when an insect inject the inactive toxin, it is converted into active toxin. Why it is converted into active toxin? Because its gut, okay, especially meat gut have alkaline pH. Okay, so this inactive toxin, Bt toxin, will now activated under the effect of alkaline pH inside the gut or a meat gut of a larva of the gut, which is solubilized the crystals. As a result of that, the crystal will be solubilized and it will be activated. The toxin bind to the surface of meat gut. Okay, especially you have to remember this thing. Okay, it will be received by the midgut epithelial cells, and it will create the pores. And finally, due to the reception of such a active toxin, it will create some pores on the midgut epithelial cells, and it causes cell swelling and lysis. Two things you will observe, swelling and the lysis, and as a result of that, cell will die and death of the insect. Ultimately, death of the insect will be result. Bt toxin genes were isolated from Bacillus thuringiensis bacteria and incorporated into crop plants such as cotton. So first of all, you have to isolate a gene which is responsible for production of this type of toxins. Okay, and that is isolated from Bacillus thuringiensis bacteria, and finally it will be incorporated, and after that means after the isolation step, it will be incorporated into the crop plant such as a cotton. Most Bt cottons are insect group specific, means it is very specific to the specific insects, such like we go through some example. The toxin is coded by the gene CRY1AC, that is the CRY1AC. It is named as a CRY. Okay, this is a crystal protein. This toxin is a crystal protein, and that's why it is considered as a CRY, CRY. In short, we can say as a CRY. So CRY1AC is responsible, and for example, proteins encoded by CRY1AC and CRY2AB control the cotton bollworm. Here you can see this is the cotton bollworm, and if you want to control such an insect, then you have to insert CRY1AC and CRY2AB inside the cotton seeds. Okay, and that of CRY1AB controls cone borer. Now if you want to control this organism, this is the insect, this is a cone borer. Okay, so if you want to control cone borer, then it is not advisable to insert the CRY1AC or CRY2AB because it is specific to cotton bollworm. So if you want to control cone borer, then you have to insert CRY1AB. Okay, this is a CRY1AB. These all kind of genes you have to select depending on your necessity. Now let me summarize whole topic by using this diagram. So first of all, what you have to do? First of all, you have to select Bt gene from the Bacillus thuringiensis, and from that bacteria you have to cut or isolate the genes by using restriction endonuclease enzyme. You know very well restriction endonuclease enzyme are working as a biological scissor. So this will cut down the Bt gene from the whole gene of a bacteria. So you have just isolated this gene from the Bacillus thuringiensis. Now you have to insert this gene inside the, you can see here, this is the plasmid. You have to insert it into the vector. This is a plasmid vector. Now by using any method, you have to insert this plasmid inside the cell of a plant. Okay, this is the host cell. So you have to insert this plasmid after you know very well competent host and all that phenomena. You have to enter this by using any method like a gene gun or electroporation or anything. Okay, so by this way this cell is now convert or GMO is formed, okay, which is now producing Bt gene. Okay, or Bt gene related product, that is a CRY protein related products. So now it become see here, this is a maize, so it is become a GMO plant, and that is a Bt maize. Okay, now what happen if you grow such a GMO plant in your farm? Then what happen if any insects are ingesting such a plant? which is already modified plant, so this insect will be killed out due to the activation of a cry protein inside their meat gut. You know very well now where it will be activated, inside the meat gut. Okay, and if you see, this is the normal plant, so it will enjoy the food. Okay, now insect will enjoy the food. But if you are modified this plant, then what happen? Paste will die when feeding on any plant part. Okay, any plant part, because whole plant is modified. Okay, whole plant is having a GMO. Okay, means this plant is a GMO and they consist this gene, and as a result of that, this gene is responsible for production of a cry protein, and that cry protein is responsible for toxic effect on the this type of insect. As a result of that, this insect will be die. Let me summarize how insect will be die. As you can see here, this insect will be ingesting this kind of a bt crop. Okay, Bacillus thuringiensis maize here it is. Okay, so if any parts of a plant is ingested by the insect, what happen? These crystals and spores are ingested by the insect larva. Okay, after ingestion, what happen? Toxins are activated to the active form in the gut, especially in meat gut, by the enzymes. Okay, and after that, these all toxin which is already activated will be received by the receptor present on the meat gut. So activated toxin bind to the receptor. Subsequently, what happen? Inserted into the membrane and cause leakage of ions and small molecule. As a result of that, pore will be generated, pore will be formed, and finally midgut membrane damage, highly damaged, and lead to starvation or a septicemia, and as a result of that, finally this insect. So this is your homework for today's lecture, and before we finish this lecture, let me summarize all topic by using one animation video. So look at this video.
Biotechnology has opened doors for many applications in the fields of agriculture, therapeutics, diagnostics, waste treatment, food processing, bioremediation, and energy production. It uses genetically modified plants, fungi, and animals to produce biopharmaceuticals. Biotechnology operates in three critical areas of research. First, it provides the best catalyst in the form of improved organisms such as microbes and pure enzymes. Second, it creates optimal conditions for these catalysts to act in. And third, biotechnology provides downstream processing technologies which help in the purification of protein and organic compounds. Biotechnology also plays a key role in agriculture and food production. With a gradual increase in the world's population, there is a growing concern about increasing the production and quality of food. Traditionally, agricultural production can be increased by employing two methods: the use of agrochemicals and organic farming. During the Green Revolution in India, crop yield had increased substantially due to better management practices, improved crops, and above all, the use of agrochemicals such as pesticides and fertilizers. The Green Revolution managed to triple food supply. However, it still fell short for the growing population. The fact remains that in developing nations like India, agrochemicals are still an expensive proposition for farmers. Besides, it is also not possible to increase the yield of existing varieties using methods like conventional breeding and organic farming. In such a situation, genetically engineered crop-based agriculture is a possible solution. Genetic engineering techniques have been used to alter the genes of many plants, bacteria, fungi, and animals, which are then called genetically modified organisms or GMOs. Nowadays, genetically modified or GM plants are being used increasingly as they have several benefits. These plants have a higher tolerance to abiotic stresses such as cold, drought, salt, and heat. And they also help reduce the use of chemical pesticides, as many of them are pest resistant. In addition, GM plants increase the efficiency of mineral usage and thereby prevent early exhaustion of soil fertility. Moreover, GM crops such as vitamin A-enriched rice enhance the nutritional value of food. Some GM plants also cater to industrial needs by providing resources such as starches, fuels, and pharmaceuticals. However, the greatest contribution of biotechnology to the field of agriculture is the production of pest-resistant plants. For example, Bt toxin produced by a bacterium called Bacillus thuringiensis has been cloned and expressed in many plants to make them insect resistant. Bacillus thuringiensis produces some crystal proteins during a particular phase of its growth. These proteins, called Bt toxin or insecticidal proteins, are toxic and inactive. Although the Bt toxin protein exists as an inactive protoxin, once an insect ingests it, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilizes the crystals. The activated toxin binds to the surface of the midgut epithelial cells of the insect and creates pores, which cause cell swelling and lysis and eventually kill the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into many crop plants such as cotton. This variety of cotton is also known as Bt cotton. The choice of gene depends on which pest is targeted. For example, a Bt toxin is coded by a gene called Cry. Cry genes are encoded by Cry1AC, Cry2AB, Cry3AB, and Cry3BB. They provide resistance to cotton bollworms, corn borer, Colorado potato beetle, and corm rootworm respectively.
In next lecture, we will learn applications of biotechnology in agriculture part 2, in which we will learn RNAi, and that is RNA interference. So till that, goodbye.